Hi, I'm Dr. Zoe Waller and I'm a lecturer in chemical biology in the School of Pharmacy at UEA. The aim of this video is to explain our recent research paper to a general audience. It's just been published in Nucleic Acids Research, which is an open access journal. This means anyone can read it, you don't need a subscription. The links to the paper are included on the YouTube webpage. The title of the paper is Identification of Multiple Genomic DNA Sequences which form i-Motif structures at neutral pH. The work is the result of a collaboration between my group in the School of Pharmacy at UEA and Dr. Julian Huppert from the University of Cambridge. So what's the paper based on? Well, our bodies are made up of many different types of cells. Within each cell is a nucleus, and this is where our genetic information is held and stored in the form of DNA. DNA itself is made up of 4 building blocks, which we call bases: A, T, C and G, also known as adenine, thymine, cytosine and guanine. In DNA, 2 strands of the building blocks can coil together to form what is known as a double helix. This structure is a bit like a twisted ladder, where the links between the strands, or the rungs of the ladder, hold them together. DNA, which is composed mainly of the bases G and C, can actually form unusual structures such as G quadruplexes and i-Motifs. These look very different to the typical double helical structure first proposed by Watson and Crick in 1953. My group are interested in i-Motifs, which form in DNA which contain lots of the base cytosine. I-Motif DNA structures have previously been used to make switches, because the DNA can fold very easily in the presence of acid to give an i-Motif structure. This structure itself is actually very tightly packed and could almost be described like a knot. But sometimes these structures can form without acid being present. So what do we already know about i-Motifs which form under neutral pH conditions? Well, first we need to think about why pH is important. So the acidity or alkalinity of a substance is measured using pH. There's a pH scale, and that runs from 0 to 14, with 0 being the most acidic. So an example household item at this end of the scale would be a car battery. pH 14 is right down the other end of the scale and is most alkaline. For example, drain cleaner is usually near about this pH. pH 7 is right in the middle of the scale, and this is described as neutral. Freshly distilled water, for example, is pH 7. In most cases, our bodies maintain a pH very close to pH 7.4, which is just on the alkaline side of neutral. When we consider what is important in biology and how things work, it is usually at or near this pH, and we call this physiological pH. So we already know that i-Motifs form at acidic pH. So I've popped one here about pH 5.5, 5.5, because this is where most i-Motif forming sequences are stable. But it's also possible for i-Motif to form at neutral or physiological pH. And this is more applicable to conditions within the body or something that would have an effect in biology. So, for example, we already know that i-Motifs can form at neutral pH under cold conditions, or when they're in crowded solution, and some sequences are actually more stable under neutral conditions than others. For example, a sequence from the hypoxia inducible factor 1 alpha gene, that's a big mouthful, is actually unexpectedly stable at neutral pH. So there are a few examples in the literature and what people have already looked at that show that these can form at neutral pH. We also know that in biology, i-Motifs can actually affect how genes are read. So, for example, normal gene expression starts at a position in the genome called the transcription start site. We know that alternative DNA structures can alter how a gene is expressed when they form in front of this region. This can essentially determine whether the gene is switched on or off. But not all genes have these sequences, and not all sequences will form i-Motif under physiological conditions. So some need acidic conditions, and some i-Motif forming sequences don't need acidic conditions. So what have we found? Well, we tested many different types of sequences that had many different numbers of cytosines in them. And we actually found that sequences with at least 4 groups of 5 cytosines are stable at neutral pH. This indicated to us that we needed a minimum number of cytosines to fold the DNA at neutral pH. This gave us an idea of what type of sequences to look for in the human genome. We found about 5,000 sequences that matched the pattern that we were looking for, and out of this, 637 of these were located in regions near transcription start sites, so regions in the DNA which were critical to whether a gene was expressed or not. So we tested 30 example sequences, and about 50% of these we found to be i-Motif at neutral pH. So what does this mean? Well, this means we've found plenty of examples of i-Motifs that are actually in the human genome and that are able to fold without using acid, so they fold at neutral pH. Many of these were found in regions of the DNA which determine whether genes are switched on or off, so they're at points in the DNA which are critical to whether something is present or expressed, or whether the gene is switched on. This work has not only given us more exciting examples of i-Motif study, but also hints at a widespread purpose for these structures in biological systems. This work's been supported by the BBSRC, the Biotechnology and Biological Sciences Research Council. If you have any questions, please feel free to ask me by either tweeting me or by email. Thank you very much for watching, and bye bye.